to read a little bit about his academic background as well. So, okay, 1 minute, please. Yeah, so Luca Peruzza is currently a researcher in zoology at the Department of Comparative Biomedicine and Food Science, that is BCA, at the University of Padova. He graded in marine biology and obtained his PhD in ecophysiology in 2018 at the National Oceanography Center, that's the University of Southampton in the United Kingdom. Studied the effect of climate change on the physiological response of decapod crustaceans to hypoxia. After his doctorate degree, he carried out a postdoc research period at the National Oceanography Center, collaborating on an international research project with the United Kingdom, India, and Bangladesh, aimed at controlling the main disease affecting carp and crustaceans aquaculture in these countries, focusing on studying how gene expression in animals was altered following infection. Subsequently, he has been a researcher at the University of Padova since 2019. His research activity is focused on understanding the ecophysiological mechanism that marine animals possess to counteract the effect of anthropogenic impacts with a focus on extreme events related to climate change, such as heat waves, integrating approaches from physiology, molecular biology, genetics, and genomics. Currently, his work is mainly focused on marine animals that are important from an aquaculture point of view, such as clam, oyster, sea bream, and sea bass. So that's just a little bit of trivia about our speaker. So my name is Emmanuel, and I'm also I happen to work alongside with Luca Peruzza, currently at the University of Padova too. So he's my boss. So thank you so much for joining us today. So I think the floor is open to you, so you can start your presentation. Thank you so much, Luca.
Okay, so thank you very much, Emmanuel, for the kind presentation. I hope you can hear me well and you can see the presentation. Okay, so let's start. I will give a brief talk about mitigation strategies against climate change in marine aquaculture, with the purpose of showcasing some of the strategies that we are currently developing in our lab at the University of Padova to tackle the threat of climate change. My talk is articulated in these sections. I wanted to start by giving a brief introduction on aquaculture and climate change, with the focus on something climate change, which is the main topic of our lab, climate extreme events. And then I will shift on the impacts of aquaculture and mitigation strategies. So I'm sure you all know that aquaculture is the controlled cultivation and farming of aquatic organisms. And these include fish, shellfish, plants, and algae, both in fresh and marine environment, freshwater and marine environment. And ideally, this practice involves breeding, rearing, and harvesting these organisms from larvae to adulthood to produce food, commercial products, and restore or enhance aquatic habitats. And as I said briefly, aquaculture can be done both on freshwater or marine environments. And if you look at marine environments, it can be practiced on coastal habitats as well as offshore habitats. Aquaculture is a vast sector, and within this sector, an important part for Italian aquaculture is related on mollusk aquaculture. And as of 2018, we had over 74,000 people employed in the European aquaculture sector. Most of them were employed in very small businesses, and but they had very relevant. They were very important in terms of enterprises, employees, and also in terms of income for the people and way of living for the people that live along the coast. What is climate change? I'm sure you all know. We define climate change the increase in human-derived greenhouse gases emissions since the Industrial Revolution. And since that period, we've been basically deploying huge tons of CO2 into the atmosphere, with the outcome that the global surface temperature have increased by over 1.5 degrees in comparison to the pre-industrial era, and will further increase much more by the end of this century, according to basically how much we are willing to reduce the emissions of CO2. And climate change is caused by humans, okay? Unfortunately, climate change, human society, and ecosystems, which are the three imaginary points in this graph, are deeply interconnected. In example, if we as human society produce greenhouse gases, that has an effect on climate change, okay, because we enhance climate change. In turn, this enhancement of climate change has dramatic impacts both on human society and on ecosystems, including biodiversity, okay? So we have to think at human society, climate change, and ecosystems as three different components of one unique system. And what we ideally want is to meet the objectives of climate resilient development by supporting human ecosystem and planetary health, as well as human well-being. This, however, requires society and ecosystems to transition to a more resilient state. And in order to do so, we have to change our habits, but also try to explore strategies that can make ecosystems more resilient. One important component of climate change is, as I said before, the increase in average temperature. The other thing that probably most of the people think of when they are asked about climate change is ocean acidification, which is the phenomena by which the CO2 emitted in the atmosphere gets absorbed in the oceans. And by doing so, sorry, is there any problem? I can hear someone. Ah, okay, no,
sorry,
that's fine.
continue. Just continue.
And the CO2 absorbed by the water results in lower in the pH of the water. And this graph on the right-hand side shows what has been the historical pH since 1850 and what will be the projected pH by the end of the century, according again to how much CO2 we are going to emit from now till the end of the century. And you might think that a drop of 0.1 in the pH scale is almost nothing, but it's actually not, because the pH scale is on a logarithm scale. So even a small change can have massive effects, particularly on species which rely on an external exoskeleton made of calcium carbonate, which, as you can see from this picture, can get dissolved very quickly if the shells are exposed to pH projected by the end of the century. However, the effect on calcifying organisms, which are typically mollusks and or corals, is not just limited to these phyla, but it's also present and very well evident for other organisms like fish. In fact, ocean acidification is able to impair the olfactory system of marine fish, rendering them less able to detect prey, less able to detect in advance the presence of predators, and making them also bold. So overall, ocean acidification has very negative consequences. The other aspect of climate change, as I said before, is the increase in water temperature. And these two graphs here show the projected production potential of marine aquaculture under climate change by the end of the century, and split by fish on the left and mollusk on the right. And as you can see, compared to the present time for the mollusk, by the end of the century, it's projected a decrease in 10% of the total surface area suitable for mollusk aquaculture, which is mostly coastal areas, okay? Again, this might seem not such a big change, but it is, in fact, because mollusk aquaculture is mostly centered on coastal areas. And if we lose coastal areas, then we lose an important sector of aquaculture and of low trophic aquaculture, because mollusks are considered low trophic species. So they are fundamental, and they will always be fundamental for providing for providing animal-rich diet to sustain the increase in our population. Sorry. And so far I've talked about ocean acidification and increasing water temperatures as two different and separate things. However, they also occur in combination. And this heat map here shows the result of recent meta-analysis with directional effects of ocean warming, ocean acidification, and the combined effect of warming and acidification on invertebrates on the left and fish on the right. And basically, how shall we look at the graph? Basically, the more red means the more negative effect on the selected process, which are listed on the rows. And whenever you find a star, it means it's a significant impact, okay? And as we can see, focusing mostly on invertebrates, ocean acidification leads to major impact in calcification, development, and growth. However, when we combine acidification and warming, the effects are even more pronounced, meaning that the combination of the two factors is not just the linear sum of the two factors. We talk about synergistic effects, in which the combination of the two gives an enhanced response, or in this case, a worsened response in animals. And this is particularly true when we consider extreme cases, okay? So extreme weather events. And climate extreme events are the main focus of the research in our lab. And we chose to focus on them because they are an aspect of climate change which up to the last decade was largely ignored. And these extreme events are defined as extreme deviations around the mean with less than 5% of probability of occurrence. And however, even if they are rare, they are increasing their frequency, their duration, their occurrence, and their intensity worldwide. And they can be extreme period of drought, in super intense rainfalls, or they can also be marine heat waves. And marine heat waves are particularly the focus of our research lab. What are marine heat waves? Are extended periods of abnormally high ocean temperatures relative to the average seasonal temperature in a particular marine region. And you might think that they are an isolated phenomenon. Well, they are not. This is the heat map showing the global ocean and the extent in terms of daily marine heatwave coverage for the year 2023. And in that year, we reached a daily marine heatwave coverage of 32%, which is about 15 times the surface of Nigeria, or 30 times the surface of France, okay? So it's not a very isolated phenomena. And in particular, if we look at the graph, let me make the pointer a laser pointer. If we look at this graph here on the top, on the bottom right corner, this graph shows the total marine heatwave coverage in terms of by year. And if you see, when we started measuring these parameter in around the 80s, we had a total marine heatwave coverage which was roughly about 60-something%. In the last years, we reached a marine heatwave coverage which exceeds by far 80%. So in 40 years, we have seen an increase of about 20% in terms of marine heatwave coverage. So they are massive phenomena. They are not small or limited, and they have dramatic repercussions. These are some of the titles which are more, those kind of titles that make up to the news, to the, first page of newspapers, like marine heat waves, climate change and failed spawning by coastal invertebrates, or mass mortality in northwestern Mediterranean rocky benty communities, effect of the 2003 heat wave, okay? So heat waves are not just phenomena that are able to cause mass mortality events, or phenomena that are able to alter the ecosystem structure or induce regime shifts in ecosystems. They also impact, sorry, fisheries and aquaculture worldwide, as you can see by these examples, in which clam fishery, shellfish fishery, finfish fishery, but also, seagrasses and corals have been massively impacted by marine heat waves. And our research at the lab of the University of Padova uses this species as a model species, which you can see here in a picture on a plate of pasta, and that's because they are delicious. And but it's not just because of that. We use the Manila clam as a model species because Italy dominates the production of Manila clam, and used to deliver up to a couple of years ago about 95% of all the European production alone, okay? And this production of Manila clam was focused on two areas which are right next door to our university, okay? And unfortunately, the production of Manila clam has, this species is an invasive species. It was introduced in the late 80s in the Venice Lagoon, where it has replaced the native clam. And since its introduction, the production of Manila clam, which you can see in this graph, has increased steadily from the 80s up to the year 2000, 2001. Then, as you can see, there has been a dramatic drop in the production of Manila clam, and this has been linked to the onset and occurrence of a very strong heat wave in 2003, which has had dramatic consequences on the production and the aquaculture of Manila clams in the Venice Lagoon, because since this event, the aquaculture of Manila clam has never really recovered. Okay, and in particular, following this event, we have observed a roughly 66% reduction of Manila clams stock. okay? And so these events are very dramatic and have very important consequences for the animals. And so they are really worth studying and to understand what are the consequences that these events are actually causing on the animals. And if we can find some mitigation strategies to ameliorate the resilience of the animals and in turn sustain the aquaculture of the species. Just give me 1 second, sorry, I have to blow my nose. Sorry. Climate change is not just okay. I can hear.
1 minute, sorry.
Yep.
Olani, Esther, can you mute yourself, please?
Okay, perfect. Sorry. Thank you, Emmanuel. Climate change is not just change in the physical characteristic of the water. Is also higher prevalence and onset of diseases, new diseases or old diseases that become more aggressive, okay? Such as the sea lice on salmon farming, which has been clearly demonstrated how the level of sea lices have increased over time, and this correlates with the rising sea temperatures. But also the increased abundance of pathogens that strike Manila clam, such as the Perkinsus olseni, which is a parasite that infects the gills of the animals. And it's not lethal in itself, but it significantly impacts the growth of the animals. And if this stress occurs at the same time with other stressors like heat waves, then animals might be more susceptible to the second stress. And also for this parasite, Perkinsus olseni, we have seen that temperature is a major component associated with the prevalence of the parasite. In fact, as you can see from the graph here, the prevalence is indicated by the red line, and we monitor the prevalence along 1 year from September to July of the following year. And as you can see, the prevalence increases quite significantly only when the water temperature starts to increase, because we are approaching the warm season. To try and mitigate the negative impacts of all the effects derived from climate change, and in particular from the effects of extreme events, we want to test and we have to test mitigation strategies, okay? Defined as any process that can ameliorate aquaculture against climate change by either reducing the vulnerability of the sector to climate impacts or by minimizing the contribution of aquaculture to greenhouse gas emissions. And these mitigation strategies include increasing animals' resilience, so acting directly on animals to make them more resilient, improving the capacity to adapt to new conditions, again, acting on animals, using probiotics, or using vaccination and or drugs, or improving feed and or farming conditions. And among all of them, we are testing in the lab two strategies, which I wanted to focus a bit more in detail right now with you. The first one is called priming. So I'm not sure if you are familiar with the concept of phenotypic plasticity. However, this picture here should make it clear. Phenotypic plasticity is the capacity of one genotype to produce different phenotypes when exposed to various environmental conditions. Okay, so think at this example as two twin plants coming from the same seed. One of them is grown in high light, one of them is grown in low light. So the two plants are genetically identical, but after some time of growth in different conditions, you might end up with this situation. One plant is very high, one plant is very low. This is an example of phenotypic plasticity, okay? The phenotypic plasticity is a big umbrella term that covers a lot of changes in all the changes in the phenotype that are not related to a change in the genotype. And among the different types of phenotypic plasticity, what we are studying in the lab at the University of Padua is called priming, which is a transient adaptation to a stress following prior exposure to a sublethal stress. Priming is also called stress memory. So basically, the simple idea is that we expose animals or plants because priming has been extensively studied and firstly characterized and described in plants. You expose the organism to a first stimulus, which is called priming stimulus, which has to be sublethal. Following the exposure to this sublethal stress, the organism triggers some adjustments at molecular, behavioral, cytological, or epigenetic level, whatever it is. It's not important whatever it is. The important thing is that the organism triggers these changes, okay? And these changes can be memorized and can be kept active for some time. This is called memory. If you then expose the same organism to the same stimulus, but this time of a lethal entity, if the memory is still active, then the organism will respond much better than a naive organism, so an organism that has not been primed. And this is what we study at the University of Padua. It's been demonstrated and proven effective in plants against heat, drought, and cold. And in animals, sorry, probably the most studied group is constituted by corals. And I said that the mechanism can be various. And this slide from a recent paper on trends in ecology and evolution is a nice diagram that shows and potentially illustrates the different mechanisms that may be taking part into the and maybe involved in priming. So we have the initial condition on the left, and on the upper boxes we have the coral host, and on the lower boxes we've got the microbiome that reside within the host. Following the exposure to priming, the animal triggers some changes, which may be at the level of the proteome by increasing the abundance of antioxidant proteins or the abundance of heat shock proteins. Okay. The host might as well act on the transcriptome or epitranscriptome by methylating the RNA, which is transcribed, but it might also act at the level of the epigenome by inducing modifications at the level of the histones, which might promote which might favor transcription or repress transcription according to the combination of epigenetic changes, okay? And following the exposure to priming, If the memory persists, then we have this condition on the up panel, okay? So the changes that have been triggered are kept, so that the animal, when the coral faces again the same stressor, the changes are already there and are there to help the coral overcome the stress. If the memory, however, is deleted, then the coral goes back to the initial stage. And if the stressor is encountered and the coral has no memory of the stressor, then we are back to the beginning, basically. And the changes on the host might as well be translated on the microbiome and vice versa, complicating further the things, okay? What we do at the University of Padova is we study priming as a mitigation strategy against marine heat waves. And in particular, we developed this experimental protocol in which we expose the animals to priming conditions, which are 1 week, constituted by 1 week at 30 degrees, which is a sublethal temperature, which doesn't kill the animals, but it's high enough to induce changes in the physiology of the animals. And then after a period of resting, we expose the animals to heatwave conditions to mimic the thermal profile of a marine heatwave, which we experienced in 2015 in the Venice Lagoon. This was an experiment which we ran 2 years ago, and we collected several endpoints during the experiment and at the end. So since the beginning of the heat wave and for the 15 days after the end of the heat wave, we monitored survival in the 4 conditions, the 2 conditions which were exposed to the heat wave and in the 2 conditions which were not exposed to the heat wave. And then we also looked at the behavior of the animals straight after the priming event and straight after the end of the heat wave. And first of all, what we saw in terms of survival was a significantly higher survival in the animals which were primed in comparison to the naive animals, which means that these naive animals are the animals that have not been exposed to priming, okay? This is the survival of the animals after during the heat wave and over the 15 days after the heat wave. And as you can see, by comparing the 2 graphs, there's a good 40% difference in survival in favor of the primed animals, okay? Meaning that priming might actually be an effective mitigation strategy against climate extremes. We also looked at the physiology of the animals, and we found out that we measured the condition index, which is the ratio of the soft tissue weight versus the shell weight. And we found that the exposure to priming has an impact on the condition index of the animals, as evidenced by the reduction in condition index in primed animals versus naive animals on the left-hand side. This group are the 2 groups that were not exposed to the heat wave. However, when we compare the 2 groups, the 2 prime groups, one exposed to the heat wave here on the right and one not exposed to the heat wave on the left, we saw no difference, okay? Meaning that the energetic debt that was paid for priming was not paid off again after the marine heat wave. On the other hand, focusing on the naive groups, we saw that following the exposure to heat wave, there's quite an important reduction in the condition index, okay? Suggesting that the animals not primed, so the naive animals, have to pay a cost to overcome the heat wave. However, the cost paid by prime animals is not paid again once the prime animals are exposed to the marine heat wave. we looked at the behavior of the animals, and these clams live within the sediment, as you can see from this video here, okay? And their normal behavior would be to hide in the sand to avoid currents and to avoid predators. What we found out was that immediately after the priming, there was no difference in the percentage of animals that were able to successfully hide in the sand within the two groups, naive and primed. As you can see, the percentages are roughly similar, 78 versus 80%, okay? Whereas when we looked at the behavior at the end of the heat wave, we found a striking difference. The animals that were primed were more able to hide in the sand than the naive animals, okay? As if the exposure to the heat wave has somehow stunned the animals, the naive animals, making them not able or less able to hide in the sand in comparison to naive animals. And we hypothesized that the cost that I was talking the slide before, the energetic cost paid by priming, also guarantees better behavior in the animals. We also looked at gene expression in two groups. The two groups of clams exposed to the heat wave, primed versus naive animals. And we found that exposure to priming upregulates the metabolism of primed animals and helps them maintaining epithelial barrier integrity. So overall, our results suggest that priming is a useful strategy that can be applied when we have to face climate extreme events. Another strategy that we are testing is focused on genetic selection. What is genetic selection? Genetic selection is the ability to boost an organism to new environmental conditions. And we do that by selecting animals that are more resilient and make population with them. We can select for many different traits, including disease resistance, thermotolerance, sex determination, growth, you name it, we can probably do it. The only thing is the trait that we are interested in must have a genetic component. Okay, so first of all, we must define a phenotype, and this phenotype must be clearly measurable without errors. And then once we have defined the phenotype, we take a population and we measure the phenotype in generation 1. After we have measured the phenotype, we can take the best performing animals from generation 1 and make a second cohort of animal using those ones as breeders, okay? If we did everything correctly and if the trait has a genetic component, the generation 2, once grown, will exhibit a phenotype which will be improved overall in comparison to the phenotype of generation 1. And the difference between the two averages of generation 2 and generation 1 is basically our breeding goal. And how does genetic selection works? Well, we want to select the beneficial gene traits from selected individuals, and we want to use those selected individuals to make the new pool of genes for the next generation. At each generation interval, we will have a genetic gain in terms of the phenotype that we are interested in. And according to the generation interval, which is linked to the biology of the organism, we might or might not have very quick selection processes. This picture here shows the average growth weight of chicken in different years. And as you can see, by selecting chicken for growth, first genetists were able to massively increase the size and the weight of the fillet of the chicken by using genetic selection. A little warning about genetic selection: not all traits are heritable, so it cannot be applied to every trait. And usually the traits that we are interested in are polygenic. It means that they are the result of many different genes which contribute a small amount to the observed phenotype. So it's a bit tricky to perform genetic selection, and there are currently three different ways of performing genetic selection: mass selection, marker-assisted selection, and genomic selection. I'm just going to briefly mention them. In mass selection, we have an initial population, and we measure the phenotype that we are interested in, and then we select the individuals which perform best, and we reproduce them. By doing so, we end up with a generation 2 in which hopefully the trait, the phenotypic trait, is improved in comparison to generation 1. Mass selection is the simplest selection technique, however, and it's cost effective. However, we have a high risk of inbreeding, and not all traits can be applied for mass selection. Luckily, there are two other genetic selection strategies based on DNA, and they mostly rely on single nucleotide polymorphism, which are single changes in one base along the DNA of the animals. And these genetic selection processes rely on the development of SNP chip, which are chips that contain hundreds, thousands of probes that are able to characterize SNPs along the genome of a target organism. And marker-assisted selection tries to link SMP data with phenotypic trait of interest. It's more accurate than mass selection, and we can select the organism also before the their phenotype is fully expressed. However, the power of the technique depends on the number of SNPs and how much polygenic the trait is. The last one is the most complicated one, and it's called genomic selection. In this method, you sequence the whole genome of each individual in the population, and you measure the phenotype as well. After you've measured the phenotype and sequenced the whole genome of every individual, you make a prediction equation, and then you can apply the prediction equation to an unseen two candidates which have not been tested for any traits that you want, and solely you can base your decision onto which animals you want to breed and which you don't, solely based on the genotype information. This is a scheme that recapitulates genomic selection. Basically, you have an initial population. Half of it are the selection candidates and are exposed to whatever you want to expose them, and you measure their phenotype. You measure their phenotype and you also sequence their genome. With those two information, you make the prediction equation, and by using the prediction equation, you then run the prediction model on the other half of the population, which has been kept safe. And based on the results, you select the animals that you want to breed, okay? This genomic selection is the most advanced technique. You don't need to remeasure the phenotype again, and helps maintaining genetic diversity and low hybriding. However, has very high costs and at the moment has limited application in aquaculture. Within our lab, we are testing two different traits: the resistance to the pathogen Perkinsus olseni in the Manila clam, and we are also testing the resistance to heat wave to lab experiments to see whether these two traits have a genetic component and if they are correlated. Because our goal is to assess whether they are heritable, and if they are, is to establish a breeding program that will help produce clam seed that is more resistant to these two threats for the Manila clam. And again, within our project, we are currently the coordinators of an European project called Shellfish Boost, in which we want to develop breeding scheme for each of these 6 mollusk species, which are the mostly farmed in Europe, and each of them has, unfortunately, its own problems. So the aim of this project, which is currently ongoing, is to tackle each of these problems in each of the species and come up with possible genomic selection strategies, or actually genetic selection strategies that can help mollusks, that can help and sustain the aquaculture sector of these mollusks. All I have said might seem a bit theoretical. I just wanted to put these genomic selection into a more concrete example. And this example was taken from finfish aquaculture, and it was found that one locus within the genome of salmon was responsible of almost all genetic variation associated to the resistance to a virus called infectious pancreatic necrosis. And by using marker-assisted selection for the favorable allele in all major breeding programs, scientists have been able to basically decrease the incidence of infectious pancreatic necrosis outbreaks basically to near to zero in just a couple of years. Okay, so if we use genetic selection or marker-assisted selection or genomic selection in an appropriate way, this is also a very powerful technique to overcome and to make organism or species more resilient to climate change. I wanted to briefly mention 2 other strategies which we currently don't work with in the lab. First one is related to probiotics, which are live microorganisms that can confer health benefits on the host. And the probiotics can be very different and can be classified according to their mode of application, via feeding on supplemented pellet, via addition to the water column, or via injection into the host. Can be differentiated according to the primary benefits, whether it's the water microbiota or the gut microbiota, and can be differentiated according to the antagonistic compound that they are going to act. And several reports have highlighted the beneficial role of probiotics, including promoting disease and stress resistance, promoting growth, improving digestion, promoting reproduction, and enhancing immunity. So also probiotics are very important mitigation strategy in aquaculture. And another strategy is related to vaccination. Vaccines are not effective, however, against climate change in itself, but can help against pathogens that are favored by climate change. However, vaccines are only for finfish aquaculture. They are ineffective for invertebrate aquaculture because invertebrates lack an adaptive immune system. So they could be powerful, but they are limited to vertebrates. And one last prevention strategy is related on early warning systems, which are systems in which, thanks to machine learning and continuous monitoring of the environment, we can detect or predict when adverse conditions are going to arrive, and we can act before the stressor or the bad conditions make a great damage onto our aquaculture facility. And that's it. There are also integrated approaches like IMTA, which in theory can make the whole environment more sustainable. However, they are still being tested. And with other partners from another project, we are also testing some miniaturized sensors to monitor stress level on almost a real-time scale, again, to have a monitoring method that can monitor stress levels and can alert us when the stress levels are getting high, so that we can intervene before the stress is too high and it has become dangerous on the animals. So I just wanted to conclude that aquaculture is currently the best solution to provide an effective source of animal proteins to meet the demands of our increasing population. Hence, we must act and we must do something to protect and safeguard this sector. Each of the strategies that I've talked about has its pros and cons. Probably the best solution is to blend the various strategies, to take the pros from each of them and try to, limit their cons. And however, the foremost goal is to slow down the progression of climate change, because, as I said before, human society, climate change, and the environment are three elements which are deeply interconnected. So with this, I'd like to thank you all for listening. I'd like to thank Emmanuel for asking me to give this brief talk. I hope it was clear, and I'm happy to take any questions.
Cool. That was I'm very interested. Make sure. So thank you so much, Luca. I mean, this is really loaded because there are a lot of gems in this 50 minutes of talking about mitigation strategies against climate change and aquaculture. So thank you so much, everyone, for joining. So if you have any question, I think there are some things you might be hearing for the first time. Understand? Probably, maybe you're in the research field or you are still a student, kind of. But I'm very sure we are kind of familiar about this climate change, even though not talking about aquaculture, we human beings we are also feeling the effect of this climate change. You understand? I remember last 2 years when I was in Spain, I was actually doing a project too on aquaculture, and probably uh there was like a very strong heat wave for like 3 days, so it really affected the species I was rearing then. So this is the right time to actually tackle it because I feel like in the next coming years it's gonna be getting worse. So thank you so much, Luca, for this amazing talk. So if you have any question, you can either raise up your hand so I can, maybe if you want to speak or you want to type it in the chat box. So, do you have any other question? Do you have questions? Paul, are you still there? I think Paul is not. So basically, if nobody has a question, I have one particular question. So
Yes.
okay, my question will just be like we are talking about climate change. And I think when talking about climate change, it's not I just want to ask, is it basically like from your lecture, I realized that you said 96% of this climate change is caused by humans, right?
Well, I mean, you said 96%.
Yeah, probably like higher percentage is caused by humans.
Yeah, I mean, it's definitely, I would say 100% caused by humans.
By humans.
The climate in the past has naturally changed. For example, there was an ice age, then there was the time of Jurassic Park when the climate was more or less 6 or 7 degrees warmer than it is today. But the difference between those changes in the past and this one is the rate of change. So we are changing the climate at a rate which is unprecedented in the history of our planet. And this is the problem because the species do not really have the time to adjust themselves. So if the change is slow, the species can adapt and have the time to overcome the change. Whereas if the change is quick and fast, there is no time for that. And then the species can either succumb or is already, in some ways prone
Declining.
to overcome, and then they survive. But most of them will succumb, will die, probably, most likely.
Yeah, that's fine. That's interesting. So,
I think
does
there was
anybody
another
ask?
question. There was a raised hand.
Oh, okay. So I'm going to read this question from Esther. It said, "What is the relevance of molecular biology to aquaculture?" Can you see the question?
Yes, I can see it.
Yeah.
Thank you, Hector, for your question. Well, I mean, the relevance of molecular biology to aquaculture is to try and find if there are markers, and by markers, I mean, for example, genes that we can use as predictors so that we can monitor those genes and see whether the condition of the animals are good or whether the animals are stressed. And this is one application of molecular biology to aquaculture, for example. And another application to molecular biology is, as I mentioned before, linked to selection of organism, genetic selection of organism, in which you really need to study the genome of the animals in order to see whether one or two or maybe more genes are linked to a particular trait that you want to maybe increase because maybe this trait is associated to higher resistance to heat or increased growth. So this is another application of molecular biology to aquaculture, I guess.
Thanks.
I think
So, yeah, okay, I can hear. I muted myself. So, thank you so much for the answer. And I think if you actually follow through the slides, you really understand that there's very big relevance in molecular biology to aquaculture because part of it is the SMPs, genetic selection, which is trying to find the best traits in maybe some particular species to actually adapt to that stress. So thank you so much for that answer. So if you have more questions, I don't know if you can raise up your hand if you want to speak. So before we close this session, because
I mean,
this
if people are shy or they,
yeah, you can put it. You can just put your question in the chat box. So
yes, or they can write me an email
yeah, exactly.
to my.
Okay, we have somebody raising up this one. So I'm going to can you unmute yourself? Ayomi Sikwa Potuje. Can unmute yourself and speak.
Maybe I can put back the last slide so you can
Yeah,
see.
the last slide. Sure, sure. Yeah, Ayomi, you raised up your hand. Do you want to say something?
How precisely do you
Everything.
mimic the heat wave process after priming the animals? Okay. Thank you for your question. We are trying to mimic the heat wave as best as we can. The heat wave that I showed you had minimum and maximum temperatures which fluctuated throughout the day from 30 to 34. And this is what we replicated in the lab to try and, of course, we cannot be we cannot realize an exact copy of what happens in the lab, because the purpose of the lab experiment is also to simplify to some extent. Otherwise, it would be easier to make an experiment in the field, but then in the field you have too many variables and you cannot generalize. So anyway, we did our best to mimic as much as we could the heat wave, so that we could get more insightful evidence as much as we could. There's another
Oh,
question.
sorry, I was trying to change my device. Can you hear me?
Yes.
Yeah, sorry. Thank you so much for the answer. Yeah, I was trying to change my device to my phone because my laptop is going off.
That's fine.
Okay, yeah. So I think
There's another
so
question
there's
we
another
have in.
question here. How do we increase animal resilience through series of research?
Well, I'm not sure I understand the question, but
Yeah, okay, yeah, probably.
I think an answer would be to testing these strategies, these mitigation strategies. May they be priming or genetic selection or vaccines, to see whether we can actually make the animals more resilient. So it's just a matter of doing research, doing it as best as we can, and also being a little bit of having a bit of luck, because sometimes also luck is important in research.
Oh, yeah. Yeah, regarding the vaccination aspect, first of all, let me ask, David, does that answer your question? Or if you want to be more, if you want it to be more clarified, you can actually always say more to. But for me, I would like to ask a question about the vaccination. I think you said it has been done in Europe, right? Is it being used in Europe? Because I feel like there are some regulations that, vaccines cannot be. I think there's a required amount or required level to be given to those fish to actually prevent them from thermal stress. Or what do you say
Yeah.
on it?
So vaccination is only valid for pathogens, and
Pathogens.
there are definitely some requirements in terms of legislation in Europe, and I'm not very familiar with those legislation, because I personally don't work with vaccines, and I've never used them. I'm sure. I mean, the vaccines that are available in Europe have been through a rigorous step of validation and everything. But I cannot really say more because I'm not really into the field.
Oh, no, that's fine. That's fine. That's fine. Thank you so much. Um, does anybody still have any other question before we close the session? So, if you want to talk or you want uh, to raise up your hands, but if
Okay.
there's no more question, I think Luca, there's to everyone listening, so there's Luca's emails is already on the
Yeah.
screen, luca.peruzza@unipd.it. So if you
Yes.
have any question, he's always welcome to answer your question via email. So
Yeah. Feel free to
yeah,
email me, and if
exactly.
you liked, you can follow us on Instagram, Facebook,
Yeah,
and all the different social medias. This is
yeah, and
uh, lab account, so you can
exactly.
the activity of the lab. We are big
And
group.
also on LinkedIn too. I think there's a page on LinkedIn too. Yeah.
Yes. So yeah, follow us if you're interested.
Okay. To everyone that joined us for this evening session, I would like to say thank you so much first to Luca for giving us 1 hour of your time and taking us through what are the current changes in climate change. Sorry, okay. And also giving us some strategies. I feel like there are more strategies, and one thing I was able to actually take from the lecture was that there's no one particular best method. So, hello, Obi, can you mute yourself, please? Yeah, sorry. So for me, I just like one thing I was able to take from today's session was that there is no particular one best approach. but the best one is to use a mixed approach. I mean, using maybe genetic selection, maybe vaccination for pathogen. So I mean, I was really able to learn a lot of things here. So thank you for your session. Thank you for taking us through the strategies. And
Thank you very
so we'd
much.
like to say thank you on behalf of Aquaworld, that I belong as a member. So we'd like to say thank you so much, everyone.
Thank you very
And
much,
thank you.
Emmanuel, and thank you very much to Aquaworld for for having me. And
Yeah, that's
I was
fine.
really glad to
Thank you so
for
much.
this opportunity.
Yeah. Okay. So take care of yourself. Ciao, ciao. Have a wonderful evening.
Bye. Bye, everyone.
Bye.